This is the, I was invited 2 years ago, and I was invited last year as well, and I couldn't make it because I think I may have been on holiday, or at a conference, I can't remember what it was exactly. But I'm going to try and give you an introduction to the immune system in my own fashion. And this is I don't really need a picture of Newcastle because we're here, so you've probably seen bits of this on the way in on trains and cars and things like that. So to start with, why do we need an immune system? Well, basically there are about 1,500 organisms out there that are out to get us, that are out to kill us, are out to cause us harm. And these range from bacteria, viruses, fungi, and even nasty worm type things that live inside us, or can do and can get inside us. So we've got all of those things that we have to battle, and we've evolved, they've evolved, and so we've got this arms race going on, and I'll come back to arms and things a little later. It's built up a little high for what's going to happen. Oh yeah, and we've got new pathogens that we're just finding every year, so as scientists we don't know as much as we like to let on. So now for graphics, and my graphics are not Hollywood, and we're not going to have big explosions, we're going to have PowerPoint slaved over there. This is a shaving accident that happens to quite a lot of us, and I think I got a bit this morning, and that breaks the first barrier of the immune system. It's our skin. Our skin is a really good barrier of defense against bacteria and pathogens of all sorts. But once it's broken, then bacteria can easily get into the bloodstream from our skin, from outside to inside, and there they'll run into complement. And we all have heard quite a bit about complement, so I won't dwell on that too much. These are some of the other barriers that we have that generally get in the way of life a lot of the time. But there again, these are things that are part of our innate immune system. So that's the immune system that is always there, ready to act. And just being a physical barrier helps to protect us. But now we come on to the active immune system. We've got these white blood cells. So we all know blood is red because of the red blood cells. There are also a lot of white blood cells in there that travel around in our arteries and our veins. Oh, here's the bacteria, they've popped up. Some have gone to the heart, they quite like to go there. A lot have ended up in the kidney, that's where they get to. By the way, this is not to scale in case you're worried. Bacteria would be much smaller and you would be able to see them, so I've just brought them up to size here. These are our white blood cells. This is a picture that I've stolen from the British Society of Immunology, of which I'm a proud member. So this is meant to be our white blood cell, and it's really good at sensing things, it's smelling things out. I don't know why that's doing it, working away there. But the bacteria give off these signals, or smells, chemicals, which these cells will seek out and destroy. And so those are neutrophils and macrophages. Those are very good, they're good at chomping, and that makes me think of Pac-Man. For all those people who know Pac-Man, maybe the younger people in the audience won't have. But I think phagocytes should be these guys. They should be a really angry, aggressive Pac-Man, because they can chomp through whole plates full of bacteria if they need to. They are collectively known as phagocytes. So just to switch my analogies and bring in my first military reference, this is the AWACS. And an AWACS is a command and control type of military vehicle. It's out there to control all the ground troops, etc., etc. And that is going to represent my dendritic cell, which is also a phagocyte, but it's not the kind of crazy attack phagocyte. It's more of a eat it and have a good think about it. And it starts to talk to its friends, which we'll come to in a minute. But it does eat and sense out these chemicals, and it's those chemicals that get the dendritic cell, or AWACS, interested. But it doesn't have much of a memory. It's a bit of a strange AWACS, but it doesn't. Its computers don't connect and it can't remember anything, so they don't remember. These are cells with memory. These are our T cells and our B cells. T cells here, which I'm going to use the stealth bomber, very sophisticated piece of kit as a stealth bomber. It can destroy things, but it's also very good at sending messages and hiding around and sneaking around and keeping an eye on things. So that's a lot like a T cell, so that's why I've chosen that. And we have our B cell, which is a bit more nimble and agile, and also is loaded full of these missiles, which are antibodies, or immunoglobulin, IG. So that's another name for that. Antibodies, immunoglobulin, we kind of use them interchangeably. I don't know why we do this in science and medicine. We make up lots of names for the same thing, and everyone gets attached to one particular one. But there we are. So this is our MIG, fighter, nimble, whatever, and it produces antibodies. That's our B cell. And of course our dendritic cell talks to these guys, and they all talk to each other through chemical signals, chemokines. So they're in constant conversation with each other, and they're organised. And because of that, these guys have memory, these ones hang around, and vaccination is based on this principle, that you put an antigen, a bit of protein into someone, and it involves the dendritic cells, the T cells and the B cells, and they respond to it, and then you have memory. They will come back and defend you again and again. So these guys can be deployed out into the field, if you like. They'll set up a little air base all over, and as soon as that comes back, as soon as you reach another pathogen comes into your system, you don't really need the AWACS. These guys will just get involved straight away. So they're your little attack force. But there's a bit of a problem when it comes to immunity that we really have to discuss, and that's the fact that how does your fighter pilot know the difference between friend and foe? How does he know that he's a long way away, it kind of looks bumpy and whatever, that could be friend or foe, I don't know, how do you tell? And that's that issue of self and non-self. We all grew from a primordial soup a very long time ago. The bacteria, us, well, we got a little bit more developed, some of us more than others. But yes, we're all from the same sort of proteins and whatever else. And so our immune system has to make that decision. Is that one of my cells? Or is that a bacteria? And for our T cell, it learns this information in this organ here, which is pretty much all this organ does, is teach T cells, the thymus. There'll be a lot of medics maybe disagreeing with that, but it learns there all our proteins, every single one of them. It tries to learn every single one of them in minute detail. And thereby, all of the ones that recognise us too much are deleted or destroyed. So we get rid of them, as many as we can. And you're left with the ones that all recognise the enemy image. So you're left with T cells that can only see these bad boys here. So that's our Japanese and our German fighters from the Second World War, I'm showing up there. But our T cells can recognise up to 1 billion foreign images. There's a number that's unthinkable. Our T cells are the same, they're trained the same way, they go to a different military academy, they do that in the bone marrow, so they're taught in very different parts of, and they're shown slightly different images, and it's a competing school. Sometimes when they come to talk together they don't talk so well, and that's where problems can happen, and we can touch on that when we come to autoimmunity. But this is how immunity works in action. We've got our bacteria, here's our green bacteria that are in the body. They run into complement straight away. There are other things here, the 3 C's, I like to think of them as chemokines and cytokines. They're similar sorts of messengers, so that's why we've got flares. They're just messengers that talk to our cells. Our B cell, because it's quite good at this kind of thing, recognises the whole bacteria in one way or another, so it can see it in 3 dimensions. The bacteria is taken up and sampled, is imaged, the scanners pick it up, there it is, there's our bacteria, the dendritic cell, who tells the T cell that it's, gives it a message that it's got this bacteria. The B cell also says, I've bumped into this bacteria, to the T cell, the AWACS, so to the stealth bomber, and that says to the B cell, yes, you need to respond, fire your missiles, and it does, and they lock on to the bacteria. And now our crazed Pac-Man comes in and goes, actually, yeah, I see it now, I hadn't seen it before, and it destroys them. And that's how immunity is working right now in your bodies. I can pretty much guarantee it. It's happening now. you're pretty healthy. There's something you've bumped into on the way here in the train. Somebody's grabbed something, you know, it's got a bit of cold, flu, whatever, and your immune system's doing that and you don't even know it. And that's the way it should be. Simple. And of course, once it's met it, it makes memory. And so it can act really quickly. Normally it would take about 8 days to get 8 to 10 days. So for that thing that you've met the first time, that's the one where you end up being sick and then you're in bed or whatever. But most of the time, it's going to happen quickly. We'll have both our stealth bombers and our MIGs, or B cells and T cells. So now we need to move, and I'll move quickly to where things start to go wrong. And mistaken identity is the clearest and most obvious thing. We all have watched our war movies. There's always a spy. There's always someone in disguise that's actually the bad guy, you know. And this is the case with a lot of bacteria, because we are so similar that some of them can actually be very close to us, and we might think, yeah, actually that is a bad guy, and so we start the whole process again, and yes, it looks bad. We've got our flares and complement, so we do make an immune response to it, and we kill it. So far, so good. But the problem is that this particular bacteria has a bit on it that's pretty similar to kidney. It's actually just a tiny little bit, and in all of this aggressive action to get rid of it, our immune systems got really fired up and a bit confused, and we start firing their missiles towards the kidney, into that thing that looks almost the same as that bacteria, to that pathogen. And now we've suddenly coated our kidney with missiles, with our own missiles, with our own antibodies, autoantibodies, with IG. It's all over the kidney. And our immune system thinks it's doing the right thing. It's attacking something that's wrong. And so of course now our pac-men, because they don't know any better, have been told to go and bind to the kidney. And so they then attack the kidney and cause lots of damage, and the whole process becomes perpetuating. So now we start to build memory towards that new protein that they moved away from the protein that was on the bacteria and closer to our kidney. For some people that won't happen. For some people they'll manage to self-correct. But for a few of us out there, we don't manage to self-correct, and we go on to build more and more of an immune response to self and have an autoimmune disease. So now I'll switch from my previous talk and move towards what we're doing now with the radar samples. So how do we detect these antibodies in that lab that is 500 yards or so, 500 meters if we want to go metric over that way? We use a thing called an ELISA, which is this is an ELISA plate, so it's enzyme-linked immunosorbent assay for its long name, microtiter plate. And we'll coat it with a target protein. Of course I'm going to go with factor H, because that's what we're really known for in Newcastle. So that's us, we've coated it onto the plate and I've washed it away. And then we put on some diluted serum. I only need about 8 microliters, or a teardrop, let's say a teardrop's worth of that to do one assay, to do one triplicate. And this patient is positive, it's bound to factor H. We need to somehow detect that, so we can use an antibody that binds the human IG. How would we get that antibody? Well, it's actually quite simple. We take purified human IG and put it into, say, a sheep, and that will make a natural immune response to that foreign antigen as it sees it, and that will make antibodies to human IG. And we can easily purify those out of the sheep. Take a little sample of blood, purify it out, then we conjugate it with this little marker here, which is going to interact with some chemicals. So we stick in our chemical messenger and it changes colour. And that's it. That's how we detect them. It's quite straightforward. We need to then throw it into a machine that we can then tell the difference between something that has colour and something that doesn't. So that's this one here down the bottom. This is actually the one I used in the lab. That's a plate. And then we get this. Data. Who's going to analyse it for me? Does anyone want to? So we get, we always set up a standard curve. It's always a little tricky. We relied on a French group who found the very first patient with anti-factor H to give us some positive sample that we could then say, okay, with this particular patient which has got a strong level of antibodies to factor H, we're going to say at that dilution, which is 1 in 50, which is where it started, then it's 4,000 relative units. And then we just dilute it out by half each time down the plate, and you get a standard curve. And we use non-linear regression to make a nice straight line, well, sort of curvy line that goes between these points. So we can take a positive patient sample here. You can see these readings of optical density, so how much blue there was. We take an average of those triplicate results, because we're scientists and we like to make sure we haven't made a mistake. We can then put that onto this curve here, which will then read us off our RU, and then you get the factor H data that Sally showed you this morning. So this is our data, this is my data. I did quite a few of these myself. We have 4 patients with really quite high titer, and we have another group of patients down here in the radar group, 7 or so patients, which make up 13% of the pediatric cohort of the children in the radar group have anti-factor H antibodies. I think it's also important to note that in our BDC, stands for blood donor control, so that's 200 people who walked off the street in Newcastle into our blood donor place and thankfully gave blood to there, and we've got a tiny little bit from that we can then use as controls. There's a couple of controls have antibodies to factor H. It's just there's a lot more in MPGN, but or C3G, DDD. There's a few more in there, so it's concentrated in there. But it is important to note that there are normal people, normal individuals who don't have any disease as far as we know, because we don't actually have a huge amount of background on them. They're healthy when they go in and donate blood. But there are some antibodies there, so that complicates our understanding of how important anti-factor H is in the disease. But it seems it is just purely because we've got 2 in 200 or 1 in 100 normal individuals versus this 13 in 100 associated with the disease. We can do a little bit more of the characterization. So if we look at factor H, it's in this kind of now stretched out in a cartoon, and it's in lots of different regions. And you don't really need to know those. You just have a bit that does the regulation part here that actually does the business function, and a bit that sticks it down to a membrane. And because we can do ELISAs and we've got lots of these little portions of factor H, we can see where they bind. And it's not a surprise to us in our field that the anti-factor H autoantibodies bind to the part of factor H that does the complement regulation. So if they bind to that part, then complement regulation is affected, and you'll get lots of C3 deposition in the kidney potentially, which is what you see in the pathology. Now, we haven't just been looking at factor H, and part of what we thought we'd do is look at some of the 50 complement proteins that are involved in the complement system. We've worked our way through, 10 different, 11 proteins of the complement system, mostly focused on the alternative pathway. Hopefully Matthew will touch on the complement system a bit more. But we have, so there's the 5 related proteins, they've got these surface regulators, and basically we haven't found much in anything else, which is a bit of it's one of the things in science, you have good ideas, hypotheses, that you're going to find these antibodies to all these other complement proteins because we've got some to factor H, and we kind of knew that that would be there, but we haven't found anything else. Except maybe 1 or 2 that have anti-C3b and factor B. So it is worth looking. We've got 1 here, potentially. These are very rare. There's one other report on the planet, I think, of an anti-factor B. There may be 1 or 2. So this could be quite interesting. With the anti-C3b, well, that's maybe not quite so interesting. It depends whether that's a true anti-C3b or a C3 nephritic factor, which is essentially the same thing. So C3 nephritic factor is an antibody to C3, the C3 convertase. So it could be some cross-reactivity there. I've got quite a bit of work to do to sort out whether it's one or the other. And I'll get to that. I will, promise. So this is two antibodies in the same endpoint. I just wanted to try and show this. So this is this C3 convertase that results in this activation of C3. So a C3 neph will bind to that, and you get lots of the C3b, which is the active C3, and that would contribute to disease. So factor H can't bind in this scenario. Factor H is a regulator of that process. We have factor H here binding to the convertase in the normal situation, so you would lose C3b. That's what you want to happen. But with an antibody to factor H, this is what could be happening. Could be happening. Notice how I've switched the language from certainty to uncertainty. But the end point is that this would be potentially important for this result that you've also seen earlier this morning, which is that you get this reduction in C3 because you get this consumption of C3 by the presence of these antibodies which bind to either the convertase or to the regulator factor H and stop it from working properly. And that also contributes to immune complexes, because what an immune complex is an antigen and an antibody bound to it, and then a few more, and then they come into, and then they settle in the kidney because of that high pressure loop that Sally introduced in the morning as well. It's a place because of that pressure that it focuses things. That's why the kidney happens to be an organ that's affected more by immune complexes than other organs. So what does this mean to you? I think I've probably covered that. Autoantibodies can associate with disease. They drive an overactivity of the complement system, but we have actually known about C3nef for, what is it, 69 wasn't it, or something? They were first reported, so 40 years nearly, and we still haven't got to the full understanding of what's going on. And say it allows us to develop theories. We love theories as scientists. We love to think about things. We love to say that we know how it works, and we go off and test it. So, theory follows. I don't know how I'm doing for time, but immunity in action part 3. So we have our kidney, and it's a B cell that's accidentally recognised it. The dendritic recognizes cell has been presented it as well, as goes, is this foreign? I don't think so. There are no flares. I'm okay. Except now factor H is disabled, so it's not working properly because it has an antibody bound to it, and we end up with now lots of complement coming around, our T cells involved, and now there's confusion in the immune system. Do I respond? Don't I respond? I've got some activation of complement. There's a signals coming, and unfortunately that leads to more cells, B cells, memory. B cells and more antibodies, which can then start to attack the kidney because the immune system has got confused by the presence of all of these messengers, all these cytokines, all this complement, and a little bit of interest in the kidney, which is actually normal for the immune system to have a little bit of interest in our organs to make sure that they're okay and they're working fine. But it does bring up the big question. Because in order for this particular theory to work, we needed antibodies to factor H. But where did those antibodies come from? Because the immune system should have been working properly before this whole started. So we have a real chicken and egg scenario going on. We know from lots and lots of research, and some that I did myself, that complement is important in forming the beginnings of the immune system, of getting rid of those cells at the academy, you know, of saying, no mate, you cannot graduate because you recognise self too much. The complement system's actually really important in that part as well, of setting the ground that we have the battle on. So any alterations in the complement system have far-reaching effects on the immune system as a whole. So it's possible that defects in the complement system lead to the antibodies and then lead to the breakdown in tolerance. I just wanted to put up one sort of slide of the kind of things that we can do once we understand we want to get rid of a B cell. We can do that now if we wanted to get rid of antibodies in B cells. And we do that in renal transplantation. And we do that in places where we know there are going to be antibodies that cause us trouble. And so we use this molecule, CD20, which is found on B cells, and we've managed to establish that it's only on B cells. And we took that and stuck it in an animal, in another animal, made that immune response happen, which would happen naturally, and collected that IG. And we've used that, reversed engineered it, and then we can put it back into people, and it will act like an autoantibody and wipe out all the B cells. So a tactical nuclear missile on our B cells. And that will get rid of them for 3 months, and that knocks down any autoantibody. So that can be used. But we've got no evidence in DDD, MPGN that we want to use it for anti-factor H or for C3NF because we don't have the right connection for that link. But they do use it in renal transplantation for that sort of thing. So we are developing the kind of weapons to deal with that. So just to summarise, DDD is a disease of inflammation on fire, and our immune system gets confused and gets activated and goes over the top, whether it's because of an infection that starts the whole process or because of something inherent, something within our genes, something that that means that our immune system is just more readily to go a little American, shall we say, gung-ho, and start attacking our own body. And when it happens, we've got lots of flares there that start the whole thing off, so we've got lots of complement, and then we have lots of involvement of the immune system, and then we go nuclear with the dropping of our 2 atomic bombs here, which wipes out the kidney with lots of collateral damage, because the immune system thinks it's doing the right thing, but it's got way off mission and way off target. Thanks for listening to part 2. I hope I haven't confused too many people.
Thank you very much, Kevin. Has anybody got any questions regarding B cells, factor H? We all know about the immune system now. Yeah? Any questions whatsoever?
Yeah.
For an insert a dummy for an antibody to attack, instead of attacking the kidney at some point, it was going to put something inside, wasn't it?
Sorry.
Doing something in Manchester.
Right. In Manchester. Yeah.